Hi class, welcome again to another session on biomaterials. So let us now discuss the membranes for plasma separation. The material that is used basically to manufacture these membranes is what we are going to discuss in today's video. So, what is first of all plasma separation? Well, the separation of impurities or harmful substances from blood is also called as plasmapheresis, apheresis, or plasma separation. So, in case of renal failure or someone with a chronic kidney disease, impurities such as urea, creatinine, and beta 2 globulin are quite high in the blood and should be removed from the blood. Now, normally the kidneys function as the filters for these material and they remove it from the blood circulation. However, if there's a renal failure, then you need a device called the dialyzer or the hemodialyzer or peritoneal dialyzer, basically depending on whichever one you're choosing to use. So, a dialyzer is required which takes over the function of the kidneys. So that would remove urea, creatinine, and beta 2 globulin from your blood. However, these are not the only impurities that are there in the blood. So you have larger molecules such as the immunoglobulin, the immune complex, the low density lipoproteins or LDL, the fibrinogen, etc. And this cannot be separated through a dialysis machine because the pore size of the membrane in a dialyzer is very, very small. So it allows smaller impurities, but it retains the larger impurities which still circulate in the blood. Now, that these are like impurities, these are unwanted stuff in the blood, and that needs to be removed. So we use a membrane to separate, which has a larger pore size that would allow these material to pass out as well from the blood. The pore size is such that it only retains the small, like only platelets cannot pass through it. All everything else is allowed to pass through it. So now the larger molecules, the molecules since they're larger than the pore size of the hemodialyzer, they're retained. And one of the main components that gets retained is albumin, and without removal of albumin or without taking albumin off blood, it is very hard to filter toxins from the blood. Therefore, you have a plasma. Therefore, in order to remove albumin, first of all you need to separate plasma from the blood, and that can be done. So plasma is basically blood minus the blood cells, so RBC, WBC, and platelets. If you remove that from blood, so whatever clear fluid remains, that is plasma for you. So this can be separate. Plasma and blood are separated either through centrifugation or through a membrane filter. So the pore size of the membrane is around zero point 0.5 microns or micrometers. So this zero point 0.5 microns can retain blood platelets, and it allows everything else to pass through it. So the plasma is then pumped in a filter that resembles a hemodialyzer, and and it migrates through the pore due to pressure difference on either side. So it is like you know um, it just acts like a filter. Either you have a pump that will suck the plasma to the other side of the membrane, or you work it through the effect of gravity. So now, the filter plasma is either treated with saline or protein solution, and then that would remove the toxin. And now, if the filtered material is like is non-usable, then that is discarded, and you inject fresh plasma from a plasma donor inside the body of the patient. Now the blood, which passes through the hemodialyzer, is then returned back without any impurities, and this is how you like purify the blood in a person with renal failure. Now plasma membranes can be of three types: the hydrophobic synthetic membrane, the synthetic hydrophilic membrane, and the cellulose-based hydrophilic membrane. So we shall have a look at each of these materials in detail now. So hydrophobic synthetic membranes are basically produced through the TIPS process, where the basic polymer PE, that is polyethylene or polypropylene, is dissolved in a hot mixture of solvents, and then it is forced through a ring-shaped spinneret. Now the solution is cooled down after exiting the spinneret, and which allows the polymer to recrystallize, and the solvent aggregates to droplets which are trapped in the polymer matrix. So now you have a material which has got which is made up of polymer, and it has got solvent material embedded inside the polymer matrix. So when you remove the solvent, then that leaves holes in the polymer matrix, and these holes actually form the pores. Now the pore formed are larger than those in blood oxygenation membrane, and therefore allow the transfer of plasma through the membrane. Now this membrane is homogeneous and has a sponge-like structure, and it has got constant pore size throughout the membrane wall. Sorry, should have gone to the next slide. Yeah. So hydrophobic plasma membrane cannot be used for plasma separation because plasma is basically quite a lot of water. 90% of plasma is water, right? So if you are having a material that repels water, then it definitely cannot filter out plasma. So you have to make sure that you hydrophilize it. You have to make sure that the membrane has affinity towards water. That's when it would invite plasma to pass through it, and that can be done by either treating the membrane surface with alcohol or solutions that contain surfactants, and then washing the membrane and the pores completely with water and saline. So you have to make sure that the membrane is not totally dry after washing this out. It has to be moist. Otherwise, if the membrane is totally dry, then that would go back to its hydrophobic characteristics, which is not what you want. If you have treated a membrane to be hydrophilic, you want the membrane to retain its characteristics, and that can be done by keeping the membrane moist. And in order to keep the membrane moist, you have to treat it with volatile hydrophilic components such as glycerol. So once you wash the membrane thoroughly to remove the alcohol or surfactant, treat it with glycerol, and then that would help the membrane maintain its moisture, and that's how hydrophobic synthetic membranes are converted into hydrophilic membranes, and then used as plasma separator. I think I have spoken enough about hydrophobic synthetic membranes in this video. I shall get back to you in another video where we shall discuss the hydrophilic and cellulose-based hydrophilic membranes. I hope you take care till then. I shall see you again. Bye.